Hello everyone, myself is Zombir Singh, Department of Geology, Government College for Women, Gohana. Today I am here to discuss about the structure, function and classification of protein. Important topic given in BSc first year, semester first, paper second. First of all, we introduce about the term protein. The term protein was first suggested by Swedish biochemist G. Berzelius in 1838 from the Greek word proteos, which proteos means preeminent or to hold first place, as these are of prime importance in structure and metabolism of cell. So, the term proteins comes from Greek word proteos. The proteins occupy the central position in the architecture and functioning of living matter. These are next to water in cellular pool and form about 12% of the cell content and 50% or more of the dry weight of the cell. Some proteins serve as important element of the body. For example, as hair, our is formed of proteins, wool, and collagen, an important constituent of connective tissues. All these are formed of proteins. Other proteins may be enzyme, hormones, oxygen carriers. Still, other proteins participate in muscular contraction, and some are associated with the gene, the hereditary factors. So, proteins are of prime importance in biological world. Proteins with catalytic activity, you can say the enzymes, are largely responsible for determining the phenotype or properties of a cell in a particular environment. The total hereditary material of the cell or genotype dictates which type of the protein the cell can produce. In fact, they have built into their structures the information that instruct them in what to do and where to go in the intracellular organization and when and how to perform. These may be activators or inhibitor. The bacterium E. coli is estimated to contain about 5,000 different type of the compound, which includes some 3,000 different kind of proteins and 1,000 nucleic acid. Along in human being, there may be 1 lakh different kind of proteins, each with unique structure. So, the separate individually each protein has the different structure. Thus, there is about 1.5 million species of the living organism on this earth. So there are probably 10 to the power 10 or 10 to the power 12 different kind of protein molecule on the living world. Proteins are polymeric biomolecules and are thus of the high molecular weight. The constituent element are carbon, hydrogen, oxygen, nitrogen, and very rarely sulfur. These are chain-like molecules produced by joining a number of the small units of amino acids called monomers or building blocks of proteins. On the basis of these introducing points, we can define the proteins as complex nitrogenous organic biopolymers of amino acids having great diversity in their organization and are of prime importance in biological world. Then next we discuss the chemical structure of protein. Chemically, the proteins are polymeric compounds formed by interlinking of amino acids. अमाइनो एसिड के इंटरलिंकिंग से बनते हैं. These amino acids are combines by equivalent peptide bonds. The amino acids are chemically formed of carbon, hydrogen, oxygen and nitrogen elements. The nitrogen element is particularly is absent from the carbohydrate and fat. फैट और कार्बोहाइड्रेट से नाइट्रोजन absent होता है. The nitrogen content of the protein is about 16%, almost 16%. Many proteins are also contain sulfur, though some proteins also contains phosphorus, iron, zinc and copper. Amino acids are monomeric building blocks of proteins. There are about 20 types of the biologically important amino acids. These are called as magic 20 in the living system. The total types of the non-amino acids is about 300. The peptide bond between amino acid is formed by dehydration synthesis or by condensation between hydroxyl group of alpha carboxyl of one amino acid and hydrogen atom of amino group of adjacent amino acid. A combination of two amino acid is called dipeptide and of three amino acid is called tripeptide, or less than 30 amino acid is called as oligopeptide. For example, glucagon of pancreas, while of more than 30 amino acid is called polypeptide. A protein may be formed of one or more polypeptide chain. An amino acid in a polypeptide chain is called as residue. The number of peptide bond in a polypeptide is one less than the number of the amino acid residue. The shape, size, structure and biological function of proteins are determined by the number, type, arrangement of amino acid, which varies considerably. This makes the protein as the most diverse type of biomolecules. Smallest known protein is thyroid stimulating hormone releasing factor of hypothalamus and is formed of only 3 amino acids. Other small sized protein are angiotensin second, formed of 8 amino acids, oxytocin of 9 amino acids, vasopressin or ADH 9 amino acids, etc. The next structure of the protein. The linear sequence of amino acid residues in polypeptide chains determines the three-dimensional configuration of a protein, and the structure of a protein determines its functions. All protein contains the elements carbon, hydrogen, oxygen, nitrogen and sulfur, and some of these may also contain phosphorus, iodines and traces of the metals like copper, zinc and manganese. The structure of protein can be divided into four levels of organization. These may be primary structure, secondary structure, tertiary structure or quaternary structure. In this structure, the amino acids are arranged in polypeptide chain in various ways. Further, linear chains of polypeptides are arranged in space in various ways. These variations in their arrangement provide the different configuration of proteins, which yield four structural models: the primary, secondary, tertiary and quaternary structure of protein. Each successive level of organization is more complex than the previous one. First of all, we take the primary structure. The primary structure of protein consists of amino acid sequence along the polypeptide chain. The primary structure of proteins includes linear arrangement of amino acid residues in a polypeptide chain. and the location of the covalent bonds like peptides, disulfide bond. If these are present, so it represents the backbone of proteins. Backbone of proteins. These are given the primary structure of proteins. It has the linear arrangement of amino acid. It is very important factor which confirms the close relationship between the amino acid sequence of the proteins and its biological function. It is the primary structure that gives a protein its physical properties and causes a polypeptide chain to hold into unique structure and acquire its characteristic function and role. A primary proteins may have any or all the 20 type of amino acid in any sequence and quantity. For example, in the primary structure of proteins, we discuss the example of insulin hormone. The first protein whose amino acid sequence forming its primary structures was determined by insulin. This protein is formed of two polypeptide chain. You can say the chain A and chain B. These chains are interlinked covalently by two disulfide bonds. A chain is formed of 21 amino acids residues while chain B is formed of 30 amino acid residues. The chain A has N-terminal glycine and C-terminal asparagine, while the B chain has N-terminal phenylalanine and C-terminal alanine. The two disulfide bonds present between the two chains, the between cysteine amino acids located at 7th and 20th position of A chain and 7th and 19th position of B chain. A third disulfide bond also occur in A chain between cysteine amino acids and the 6th and 11th position. Both chain of insulin are biosynthesized as single polypeptide chain proinsulin in which A in which chain A and chain B are interlinked by connecting polypeptide of 33 amino acids. Its synthesis is controlled by gene located on the short form of chromosome length. It is then undergoes proteolytic processing forming insulin. Primary structures of ribonuclease was determined by Hayes, Murray and Steen 1960. It is consisted of single polypeptide chain of 120 amino acid residues with lysines at N terminal and valine at the C terminal. So the primary structure determines further the level of organization of protein molecule. Then next we discuss the secondary structure. The secondary structures includes various types of the local conformation in which atoms of the side chains are not involved. Atom of the side chains are not involved. The secondary structures refers to the conformational arrangement of amino acids in the polypeptide chain shows the steric relationship between them. It indicates the bending and folding of the polypeptide chain. So you can see that the secondary structure of the proteins explains the bending of the linear arrangement of the polypeptide chains which is given in the primary structure. Louis Pauling and the Corey 1951 demonstrated the secondary structure of the proteins in the following way. First point, alpha helix secondary structure, and second is the beta helix secondary structure. Alpha helix secondary structure. The alpha helix is right-handed coiled strand. Alpha helix, it is characterized by a polypeptide chain is spirally coiled to form a three-dimensional form along the longitudinal axis of the molecule, so provides a condensed structure. Second, the side chain of amino acid protrudes outward from the helical backbone. Third is each spiral is completed in about 5.4 angstrom and has a diameter of 6 angstrom. The fourth point in the structure of the secondary alpha helix conformation is stabilized due to the presence of a series of regularly spaced intramolecular hydrogen bond between every imide hydrogen and the electronegative carbonyl oxygen of the fourth amino acid behind it in the helix. So each peptide bond of the chain participate in the conformation of the such hydrogen bond. Thus, amino acids spaced third or fourth apart in linear sequence are spatially closer to one another in a alpha helix, as intramolecular distance is 1.5 angstrom. So on average, each helix contains about 3.6 amino acids. Alpha helix forms is the most stable conformation for a polypeptide chain. Hydrogen bond forms between the oxygen of the carbonyl of each peptide bond in the strand and the hydrogen of the imide group of the peptide bond for amino acids below in the helix. The two strands can be either parallel or anti-parallel depending on whether the strand direction and terminus to C terminus are same or opposite. The anti-parallel base alpha helix conformation are found in alpha keratins, which are of major component of the exoskeleton structure of vertebrate. Hair, wool, feather, nail, claws, scales, horns, tortoise shells, and stratum corneum of skin. It is found in both fibrous and globular proteins. Further, alpha keratins are rich cysteine residues which forms disulfide cross linkage between the adjoining polypeptide chain. These bonds are very strong and provide great cohesive strength. Then next we discuss the beta plated seed structure. Beta plated seed structures is an extended structure in which the protein formed of a number of polypeptide chain lying side by side. In these adjacent polypeptide chains run either in opposite directions nitrogen to carbon terminus called anti-parallel beta plated seed, or in same direction called parallel beta plated seed. The amino acid in a polypeptide chain are spaced at 3.5 angstrom. Amino acids are rotated through 180 degree with respect to adjacent amino acid residues. There are intermolecular hydrogen bond between imide hydrogen and carbonyl oxygen of adjacent polypeptide chain. Maximum hydrogen bonding is possible in antiparallel structures. There are no cysteine residues, so There are no disulfide linkages between the adjacent polypeptide chain. Beta plated structure is found in fibrous proteins of the silk fiber. It has repeating sequence of 6 amino acid residues: glycine, serine, glycines, alanines, etc. It is also found in spider web fibers, synthetic fibers like nylon, orlons, and antibodies. In certain proteins, bovines, pancreatic ribonuclease, both alpha helix and beta plated sheet structure are present. This proteins forms the alpha helix formed in one part by coilings, and plated sheet forms in another part by bending on itself. Then next we discuss about the tertiary structure of protein. Tertiary structure refers to the mode of bending or folding of polypeptide chain to form a compact, tightly folded and three-dimensional globular structure. It is characterized by a polypeptide chain takes three-dimensional shape, so is either spherical or globular or irregular. It is also compactly folded, that there is little or no space for water molecules. The chain is not arranged along one axis but is irregularly folded. So the tertiary structure of proteins is produced by interaction between amino acid residues that may be located at a considerable distance from each other in primary sequence of the polypeptide chain. Hydrophobic amino acid residues tend to collect in the interior of the globular proteins, whether they exclude water, whereas the hydrophilic residues are usually found on the surface where they interact with the water. Tertiary structure is found in all enzymes, proteins, proteinaceous hormones. The globular proteins with well-known tertiary structures are myoglobins, lysozymes, carboxypeptidase, cystochromes, C, lactate, dehydrogenase, etc. All these compounds contain the tertiary structure of proteins. Tertiary structures of sperm whale myoglobins was given by J.C. Kendrew et al. 1960. Then we discuss the quaternary structure of protein. Quaternary structures refers to the interaction of one or more subunits to form a functional proteins using the same forces that stabilizing the tertiary structure. It is spatial arrangement of the subunit in a proteins that consists of more than one polypeptide chain. Such proteins are called oligomers or multi-subunits proteins, while the individual polypeptide chains are either protomers or monomers. The number of polypeptide chain involved can be determined by determining the number of amino acid residues per protein molecule. Each protomer has its own characteristics, secondary and tertiary conformation, but the protomers can take different spatial conformations with relation to another. These changes are responsible for change in the properties of proteins. These protomers may be same, then called homogeneous quaternary structure. For example, the enzyme phosphorylase but may be different than called heterogeneous quaternary structure. For example, tobacco mosaic virus or and insulin. So these are the two examples. The forces which stabilizing the quaternary conformations are hydrogen bond, hydrophobic interactions, and electrostatic or the salt bond between the residues on the surface of polypeptide chain. First oligomeric protein studied by X-ray diffraction studies was hemoglobin, which shows a variety of conformations depending upon the oxygenations. It is tetrameric proteins formed of the four polypeptide gene and the four heme prosthetic group, each having iron in the ferrous state. Then next point, we study the cotton. We study the properties of proteins, solubility in water. Relationship of proteins with water is complex. The secondary structure of the proteins depends largely on the interaction of peptide bonds with water through the hydrogen bonds. Hydrogen bonds are also formed between the proteins alpha and beta structure के अंदर and water. The protein rich static ball is more soluble than the helical structures. At the tertiary structure, water causes the orientation of the chain and hydrophilic radicals to the outside of the molecules, while the hydrophobic chains and radicals tend to react with each other within the molecule. The hydrophobic effect. Denaturation and renaturation. Proteins can be denatured by agents such as the heat, urea that causes unfolding of polypeptide chain without causing hydrolysis of peptide bond. So, we can define that denaturation as the process which involves the unfolding of polypeptide chain due to the reaction of due to the heat or the reactions with other chemicals. Denaturation agents destroy the secondary and tertiary structure without affecting the primary structure. If a denatured protein returns to its native state after denaturing agent is removed, the process is called renaturation. Some of the denaturing agent includes physical agent, heat, radiation, pH, chemical agent urea solution which form a new hydrogen bond in the protein, organic solvents and detergent. The next property is the coagulation. When proteins are denatured by heat, they form insoluble aggregates known as coagulums. All the proteins are not heat coagulables, only a few like albumin globulins are heat coagulable. The next property is the isoelectric point. The isoelectric point is the pH at which the number of positive charge equals the number of negative charge and the overall charge on the amino acid is 0. At this point, when subjected to an electric field, the protein do not move either toward the another anode or cathode. Hence, this property is used to isolate protein. Molecular weight of the protein. The average molecular weight of an amino acid is taken to be 110. The total number of amino acid in a protein multiplied by 110 gives the approximate molecular weight of that protein. Different protein have different amino acid composition and hence their molecular weight is different. The molecular weight of the proteins ranges from 5,000 to 10 to the power 9 daltons. It occurs after proteins has been synthesized on the ribosome. Chemical properties. Biuret test: when 2 ml of the test solution is added to an equal volume of 10% sodium hydroxide and 1 drop of the 10% copper sulfate solutions, a violet color formation indicates the presence of the polypeptide linkage. These are the tests for protein. Then second is the ninhydrin test. To perform the ninhydrin test, we take 1 ml of ninhydrin solution is then added to 1 ml of protein solution and heated. Formation of a violet color indicate the presence of alpha amino acid. The next point is the classification of the proteins as given in this hierarchy based on structure and based on composition. Based on structure, the proteins are of three types. First is fibrous proteins, globular proteins, and intermediate proteins. Based on composition, simple proteins, conjugated proteins. The simple proteins may be albumin, globulin, histones, scleroproteins, and conjugated proteins, phosphoproteins, glycoproteins, nucleoproteins, chromoprotein, lipoproteins, and flavoprotein. Then we discuss their classification one by one. First is the fibrous protein. Fibrous protein. In these proteins, the polypeptide chains are extended along one axis and are spirally wound to form fiber. The example are alpha keratins of hair, nails, claws, horns, hooves, scales, feather, fibrins of silk fiber, collagen of tendons, elastin of ligament, myosin and actin of muscles. Then globular proteins. In globular proteins, the polypeptide chain are compactly folded to form the compact spherical or globular shaped proteins. These are the globular proteins. Example are egg albumin, serum albumin, globulins, hemoglobins, all enzymes, proteinaceous hormones like insulin, venom of snake, and scorpion all are globular proteins. Then we come on the second point, simple protein. Albumins. Albumins is soluble in water, dilute salt solution, and dilute acid and bases. These coagulate on heating and precipitate with high salt concentration. The example is egg albumin, serum albumin एंड लेक्टाबुमिन्स मिल्क के अंदर होता है जो लेग्यूमलेंस जो सोयाबीन के अंदर होता है. देन नेक्स्ट इज द ग्लोबुलिन. ग्लोबुलिन इज insoluble in water but freely soluble in dilute salt solution. These are precipitated by the half saturated salt solution. Example is serum globulins, vitellins, tuberins, myosinogen. Then next is the histones. Stones are soluble in water and salt solutions but insoluble in very dilute ammonium hydroxide. Example, globin, hemoglobin. Globin protein is found in hemoglobin. Basic proteins with nucleic acids in chromosomes. Scleroprotein. Scleroproteins are insoluble in water, dilute acid and alkalis. It is resistant to digestive juices. Example is keratin of hair, nail, clutch, hoof, horn, feather, fibrous fibroin proteins of silk fiber, collagen of tendon, elastin of ligament. Then next we come to the conjugated proteins. Phosphoprotein. Phosphoproteins is associated with phosphoric acid residues. Examples are the casein of milk. Then second is the glycoproteins. Glycoproteins is the combination of the proteins with mucopolysaccharides. Example is the mucin in saliva, blood antigen, pituitary hormones like follicle stimulating hormone and luteinizing hormone. All these are the example of glycoproteins. Nucleoprotein. Nucleoproteins are the basic proteins. These are the basic proteins plus nucleic acids. Example, deoxyribonucleoproteins in chromosomes of nuclei and ribonucleoproteins in ribosome. Then next is chromoprotein. Chromoproteins associated with a certain porphyrin ring pigment. Example, hemoglobin, myoglobin, cytochromes, rhodopsin, flavoproteins. All these are important example of chromoproteins. Lipoproteins. Lipoproteins are formed by the combination of proteins with lipid. You can say the fatty acids, cholesterols, or the phospholipids. The important examples are egg yolk, serum, milk, cell membranes. Then next is the flavoprotein. Next we discuss the functions of protein. First, the protein functions as enzyme. So all enzymes are proteins. Second is the structural proteins in animal form the structural material other than the inorganic component of the skeleton. Second, the storage proteins can store small molecules or ion. Protective proteins are antibodies, a substance that helps protect the body from invasion by virus, bacteria, and other foreign substances or the pathogens as well as the blood clotting. Regulatory proteins are responsible for hormone regulation of body processes. Nerve impulse transmission proteins are receptors of small molecules that pass between gaps or separating nerve cells. Movement proteins are muscle proteins which are found in the muscles. The transport proteins are proteins that bind and transport numerous small molecules and ions through the body. So the proteins are of prime in function.